A very good morning to all of you. So today's our topic of discussion is that of OXPHOS disease. OXPHOS disease asked as a short note. Individually it can be asked as a short note, or question can be asked in the, if LAQ will come, then question can be asked in the form of that, what is electron transport chain, what is chemiosmotic theory, write the inhibitors and uncouplers of ETC, along with explain the OXPHOS disease in brief. Okay, if LAQ will come, then it can be asked in that way. Otherwise, simply short note can be asked in the OXPHOS disease. It's important topic. Okay, so let's start the topic. We will understand that what is OXPHOS disease. So OXPHOS disease is also known as mitochondrial disorder. Okay, mitochondrial disorder. So either OXPHOS disease will come or question can be asked as a mitochondrial disorder. So both are the same thing. So mitochondrial disorder and OXPHOS disease are the same thing. So what are OXPHOS disease? OXPHOS, as the name suggests, these are the disorders that are related with your oxidative phosphorylation. Ox means oxidative, phos means phosphorylation. Okay, so from oxidative phosphorylation, this term OXPHOS will come. So what are OXPHOS disease? These are what? These are disorders related to oxidative phosphorylation. These are the disorders which are related to your what? These are related to your phosphorylation. Okay, so first thing is these are the disorders which are related to your oxidative phosphorylation. Okay, so how this disorder will cause? It is caused by what? It is caused by due to mutation in what? It is caused by due to mutation in your mitochondrial DNA. Okay, mitochondrial DNA. As you have studied before, that the mitochondria has its own DNA. Okay, as like in cell, what inside cell there is a nuclear DNA which is present inside the nucleus, and there is also in mitochondria has its individual DNA that is known as your mitochondrial DNA. Okay, so these disorder, how these disorder will occur? These disorder will caused by mutation. When the mitochondrial DNA is mutated, then it will cause your OXPHOS disease. Okay. Next thing is that mitochondrial DNA. The question is asked in the Viva also, why mitochondrial DNA is more prone for the mutation as compared to your nuclear DNA. Okay, so what is the answer? The mitochondrial DNA is more prone. Mitochondrial DNA is more prone to mutation. Mitochondrial DNA are more prone to mutation as compared to nuclear DNA. As compared to your nuclear DNA. The question is, mark is there. Why? So reason is that, why mitochondrial DNA more prone to mutation as compared to your nuclear DNA? Because there is no DNA repair mechanism. Okay, because there is no DNA repair mechanism. Got it? No DNA repair. So if mitochondrial DNA is once mutated or damaged, then there is no DNA repair mechanism, as we have seen in case of nuclear DNA. So once they are mutated or once they are damaged, then there is no mechanism by which they will come back in their original form. That's why mitochondrial DNA are more prone for mutation as compared to your nuclear DNA. Okay, so this question can be asked as in Viva, and it is important as a MCQ also. So you have to remember that because of no DNA repair mechanism, mitochondrial DNA is more prone for mutation. Okay, so this is the in OXPHOS disease. What is? These are related to your oxidative phosphorylation, and these occur due to mutation in mitochondrial DNA, and mitochondrial DNA is more prone to mutation because of your no DNA repair mechanism. Next point is mitochondrial DNA has what kind of inheritance? Mitochondrial DNA has your maternal inheritance. Okay, maternal inheritance is found in mitochondrial DNA. Okay, M for mitochondria, M for maternal. So always remember mitochondria follow your maternal inheritance. That's why if any male is suffered from this OXPHOS disease, they cannot transmit it to your further generation because these are only maternally inherited. Okay. Next point is which two organs are more affected by, or which organs are more affected by this OXPHOS disease? So your CNS and your heart muscles. CNS and your heart muscles, your CNS and your heart muscles are vulnerable. These are what? These are your vulnerable. Vulnerable for OXPHOS disease. Why? Because they are more dependent on, because these two organs are more dependent on your oxidative phosphorylation. Oxidative phosphorylation. Got it? So these points first you will mention as an introduction, that is what is OXPHOS disease or what are mitochondrial disorder. So OXPHOS disease are the disorders which are related to your oxidative phosphorylation, and these disorders occur due to mutation in your mitochondrial DNA, and mitochondrial DNA is more prone for mutation because there is no DNA repair mechanism. And next point is mitochondrial DNA follow your maternal inheritance. Okay. Next, your CNS and your heart muscle are more vulnerable for OXPHOS disease because they are mostly totally dependent or more dependent on your oxidative phosphorylation. Okay. Now let's understand what are the disorders or what are the defects which are caused in your OXPHOS disease. So you will make it's a table. You will write it in a tabular form so that it's easy for you to write and it's also easy for the examiner to check your copy. Okay, so let's make a table of all the kind of your OXPHOS disease. So first you will write the defect. First table you will write the defect and then cause. What are the cause of that defect? Okay, and then your clinical features. Okay, so first what are the defects? What is the name of defect of that OXPHOS disease? What are the causes and what are the clinical features? So first is your that of LHON. What is the full form? It's a Leber's hereditary optic neuropathy. So first defect is that of, in short form we write all Lohan, LHON. It's a Leber's hereditary optic neuropathy. Okay, first OXPHOS disease. What is the cause? Cause is due to defect in mitochondrial DNA gene of your complex 1 in ETC. When your complex 1, which is the complex 1 in ETC? That is your NADH coenzyme Q oxidoreductase. Okay. So when there is a defect in your complex 1 of your ETC, then it will cause a defect that is known as Lohan, LHON, that is Leber's hereditary optic neuropathy. So what clinical feature you will get in it, as this is optic neuropathy, that means it will affect which organ? Optic, that means eye is affected, neuropathy, your brain is affected. Okay. So what feature? Patient present with blindness and patient present with tremors, ataxia and seizures. Okay. So these are the clinical feature you will get in Lohan. Now comes second disorder. Second is that of MELAS. MELAS. What is the full form of it? That is mitochondrial epilepsy, lactic acidosis and stroke. Okay, so what is the full form of MELAS? What is MELAS? MELAS. Okay, MELAS, MELAS. So mitochondrial epilepsy, L for lactic, A for acidosis, S for stroke. Mitochondrial epilepsy, lactic acidosis, and stroke. Second disorder. Why this occur? This occur due to mutation in mitochondrial DNA gene that affect your tRNA, that is transfer RNA, okay, tRNA in amino acid leucine. Okay, when the leucine amino acid tRNA is defective, then it will cause your MELAS. Okay, so what is the clinical feature? As it show mitochondrial epilepsy, lactic acidosis, and stroke. As the name suggests, so what will be the feature are there? So clinical feature will be stroke is there. Okay, stroke is there. Then your lactic acidosis will be there. Stroke is there, lactic acidosis, and so lactic acidosis will be there, stroke is there. Stroke, STR, stroke. Epilepsy, so patient present with your what? Seizures and your epilepsy. Okay, seizures and your epilepsy. Okay, so these are the clinical feature you will get in MELAS. Stroke, lactic acidosis, seizures, epilepsy, and sometimes diabetes mellitus is also there. Alright, so these are the clinical feature of your MELAS. Now third is that of MERAF. MERRF. Its form is that of myoclonic encephalopathy. Myoclonic encephalopathy and red fiber. Okay, so myoclonic epilepsy, red fiber. RAGD, it's a RAGD, RAGGED. Myoclonic encephalopathy and RAGD, RAGGED, RAGD, red fiber. Okay, so what is MERAF? That is myoclonic encephalopathy, RAGD, red fiber. Okay, why does disorder occur? It occurs due to mutation in your mitochondrial DNA, and which it affects particularly, which it's unknown. Okay, when you will read most of the books written, it is on the cause is only somewhere there is a mutation in mitochondrial DNA. Okay, nothing more that in this for this disorder. So what is the clinical feature? Clinical feature is that of seizures will be there, encephalopathy, so seizures will be there, tremors will be there, and there is a lactic acidosis is also found in this disorder. Lactic acidosis, and on muscle biopsy, one of the specific feature is that of that when you will do the muscle biopsy, then get RAGD red fiber. Red fiber. Okay, so this is the diagnostic of this MERAF. First you will get, as this is encephalopathy, so as usual you will get seizures, tremors, ataxia, tingling, numbness, and lactic acidosis there. And when you will do the muscle biopsy of patients suffering from this, then you will, on muscle biopsy, you will get your RAGD red fiber. Alright. Now fourth is that of leak syndrome. LEIGH apostrophe S. Leaks syndrome. Leaks syndrome. LEIGHS. Leaks syndrome. Leak syndrome. What is the cause of this leak syndrome? It is due to mutation in your complex 1 also. Mutation in complex 1 of ETC. Okay, so remember, in complex 1 mutation, it will cause 2 disorder. One is that of Lohan and other is that of Leaks, LL. Both LL disorder are caused by the mutation of the complex 1. And Leak syndrome, the clinical feature is that of there is a movement disorder. Movement disorder. Okay, so these are the four important disorders of your OXPHOS disease, and in the tabular form you can write in your exam, you will get a good marks. Okay, so what is the first defect? That is of LHON, that is Leber's hereditary optic neuropathy. What is the cause of this defect? This occurred due to mutation in your complex 1, that is NADH coenzyme Q oxidoreductase is the complex 1 of ETC. Leber's hereditary optic neuropathy, that will affect your eye and it will affect your brain or CNS. So features will be present because of affecting eye, blindness will be there, and because of affecting your CNS, tremors, ataxia and seizures will be there. Second is that of MELAS. What is MELAS? That is of mitochondrial epilepsy, lactic acidosis, and S stands for stroke. What is S here? S stands for your stroke. So the full form is that of MELAS, MELAS. Okay. So why this disorder will occur? This occur due to when the tRNA which code for your leucine amino acid, that tRNA gene is defective. Because of that defective gene, ultimately your leucine amino acid will be defective and it will cause your MELAS. So as name suggests, mitochondrial epilepsy, lactic acidosis, stroke. So you can easily write the features, clinical feature, lactic acidosis will be there, stroke will be there will be epilepsy, seizures, and sometimes diabetes mellitus is also prone in these patients. Okay. Next is that of MERRF, MERRF, that is myoclonic encephalopathy and ragged red fiber. And what is the cause? Cause is known, somewhere it is due to mutation in mitochondrial DNA. And as being an encephalopathy, so patient present with your seizures, patient present with your tremors, and lactic acidosis will be there. And diagnosis, how you will diagnose this? So when you will do the muscle biopsy, you will get on muscle biopsy, there is a ragged red fiber. Okay. Now fourth disorder is that of Leak syndrome. Leak syndrome is also due to mutation in your complex 1 of your ETC, and here patient present with your movement disorder. Okay, so this is all defect which occur in your OXPHOS disease. Now comes next, you will write down diagnosis. How you will make the diagnosis of this OXPHOS disease. Okay, so how you will make diagnosis? So diagnosis you will do, first diagnosis you will do the muscle biopsy. For diagnosis you will do muscle biopsy, and second you will isolate mitochondria. Okay. For diagnosis, what you will do? First you will do muscle biopsy, and if you didn't get anything, then you will isolate mitochondria, and you will see by various method of molecular technology, you will see which gene is defective. Alright. Or now, how you will treat patient? So there is no specific treatment. No specific treatment. And simply treatment is based on symptomatic. So simply you will treat the patient according to that symptom. So no specific treatment for this disease, simply you will do symptomatic treatment. Alright. So this is all about your OXPHOS disease, must read topic and important topic for short note, and sometimes included in LAQ. So this is a small topic, you can easily read out. Thank you.